myself. And our next speaker is Erik Ciaccio. Erik, you may share your screen. And he's going to talk about cryptic diversity and the importance of biogeography, preliminary results in a genus of mygalomorph spiders from the Pacific Northwest. Okay, we can see your screen and you may now begin at any time you want. Thank you.
Hi, everybody. Thank you for coming. My name is Erik Ciaccio, and I'm a PhD student at the University of Idaho. And today I'm going to be sharing some preliminary results that I have with a genus of mygalomorph spider, Antrodiaetus. So Dobzhansky has famously stated that nothing in biology really makes sense except in the light of evolution, and that evolution is this central theory that underpins biology. And so what evolution is to biology, species are to evolution, as they're this foundational unit that kind of unites this theory. What are the taxa? This is a core assumption in evolutionary biology that we make when we ask questions. And thus it's a basic piece of information that we need to possess for biological questions. Following this logic, an accurate understanding of what the taxa are, this is required for asking evolutionary biology questions without injecting systemic bias into the framework. So this need to accurately understand the taxa in our system inevitably leads us to a point where we sometimes must delineate the boundaries between different lineages in order to have a complete picture. But this can be difficult when lineages consist of cryptic species. So cryptic species are two or more distinct lineages currently incorrectly classified as a single lineage, due in part to conserved or confusingly convergent morphologies, which confound our ability to correctly discern taxonomy. So the study of cryptic species of themselves is interesting because they are such a challenge to our approaches in species limitation. But aside that pure pursuit, they're also important as they represent unaccounted diversity and hint at broader biological processes such as niche conservatism. So whether the questions we ask are centered around biodiversity or biology more generally, cryptic species are this potential source of systemic bias in our analytical frameworks that needs to be dealt with. My research is focused on Antrodiaetus, more commonly known as folding trapdoor spiders. This is a genus from the family Antrodiaetidae, and it's a relatively old lineage of spider. They fall within the larger Adipoidea group, which is sister to the rest of the mygalomorph spiders. They're pretty deep at the root of the spider tree of life. They're very morphologically conserved. If you go out into the field and you look for some of these trapdoor spiders, they're more or less going to look like this. There's subtle morphological differences that have been used previously to tell the species apart. But as I said, they're rather conserved when it comes to morphology. In fact, cryptic species have been discovered in some of the eastern taxa of Antrodiaetus. They're burrow dwellers and ambush predators. These guys will make small little burrows going into the hillsides of the landscape they're in, and they'll pretty much sit in those burrows and wait for prey items to come by, or they'll then just reach out and grab them. They're often range restricted to specific habitats depending on the species you're looking at. And they're rather dispersal limited. These spiders, for the most part, will not move throughout their entire lifetime. Once they make a burrow, unless they're chased out of it or forced to move, they're not going to relocate. The only exception to this is during the mating season when males will come out of their burrows and wander looking for females. But other than that, after that kind of initial natal dispersal by spiderlings, and they do not balloon like many other araneomorphs do. All that dispersal is based just by walking. They're really not going to go very far. And so what all this means is that Antrodiaetus evolution is often a reflection of geological history. And so the study of Antrodiaetus can be really informative to understanding larger biogeographical pictures of entire regions. So if we look at where Antrodiaetus is distributed, they have this whole Arctic distribution, with most species occurring in the Nearctic. There's 2 species currently recognized in Japan. In the Nearctic, those species are split between the east and the west, with most of the species occurring in the northwest. So this that region up here in green with the box. This is what I'm going to be focusing on for my research, and it's what I'm going to be focusing on for the talk today. Looking at previous work that has been done with those Western Nearctic Antrodiaetus, most of the work was based off of, most of the phylogenetic work, I'm sorry, is based off of morphological and limited use of mitochondrial and nuclear markers. To the important paper or pieces of work that I'm using here, or as a springboard for my own research, is that of Miller and Coyle from 1996 and Hendrickson Bond from 2007. Now, one of the things that's important to note about these topologies, these relationships that they've reconstructed, is that they're in confliction with one another. Those two works don't necessarily agree on how all the taxa are related to one another. It's also important to note that they found this kind of lack of geographical cohesion among regions. So when you're dealing with these kind of poorly dispersing animals, you expect relationships to more closely map geography, but that's not the case. So here I've circled in green the taxa from the Northwest region, and as you can see, they don't form a clade here. And these previous works have also discovered paraphyly in certain taxa. So specifically, Antrodiaetus pacificus, you can see here is paraphyletic, with Antrodiaetus apachicus kind of breaking them up. And this is a taxa from the Southwest, from the Sky Islands down in Arizona. So looking again more closely at those Northwestern Antrodiaetids, currently there are about 10 recognized species from the Northwest. And they occupy a diversity of habitats here. So you can find them in habitats such as those mesic montane slopes you might see in the conifer forests of the Rocky Mountains or the coastal ranges, down into some of the more drier xeric high plains sage scrub regions. And it's really going to be species specific. What is important to note is that most of the work does not include the full range of these species. So for example, here in red, I've circled collections that were used in Hendrickson Bond 2007. This is Antrodiaetus pacificus. But if we look at something like iNaturalist, we can see that there's far more observations than what is currently represented in the literature. So they're more broadly distributed than we've kind of described in the literature. This is an extremely large range, talking about just Antrodiaetus pacificus. For an Antrodiaetid, this is very large. It's several times larger than the other taxa in the region. And it's also geographically split between this western group and this eastern group, and it's split by the Columbia Plateau. Now, this is interesting to note because Antrodiaetus pacificus occupies these kind of montane habitats, these mesic montane habitats where they're a little bit more moisture dependent. You can see here up top, this is a picture of one in their burrow, this kind of clay-like damp soil. The habitat here is closed in, it's shaded. The Columbia Plateau is, as the name suggests, this plateau. It's made up of grasslands. The region of the Palouse is located here. There's a lot of farmland out here. And this is probably representing some strong ecological barrier to movement between these populations. So some of the questions I have about Antrodiaetus pacificus specifically is just how large is that range? So we've seen observations going all the way up into Canada, but are those actually Antrodiaetus pacificus? Are there multiple distinct lineages within Pacificus, as has been hinted at, has been seen in previous works? Are those really distinct lineages? And if yes, what forces might be responsible for creating those distinct lineages? So currently, I have a genomic data set that consists of several of the taxa from the Pacific Northwest. Looking at Pacificus again specifically, I have both that Western and Eastern population represented in that data set. I haven't done any sampling yet. This is to be done, hopefully later this year in Canada with those northern populations. That is planned. And so that genomic data consists of UCEs, or ultra conserved elements. What these are a type of genomic marker that is represented by this core conserved sequence surrounded by an increasingly variable flanking region. I used the spider-specific probe set from Kulkarni et al. 2020 to extract those UCE markers. And my data set consisted of almost about 500 different loci. So to give you an idea of what these UCEs look like, down here I have an alignment. So this would be an example of a single UCE locus. I have an alignment here with this core conserved region here where there's not much variability. But as you go out to those flanking regions, you get an increasing level of variability among taxa. And so this is useful for looking at various depths into a tree. Over here is the preliminary reconstruction of those Northwest Antrodiaetidae that we're going to talk a little bit about. So to put that in perspective here, again, if we just focus on Antrodiaetus pacificus, we can see that there is this Western and Eastern population, and we can also see that they're paraphyletic. So here in blue, I've represented the Western Pacificus, and here in purple or pink, I've represented the Eastern Pacificus. You can see that they are paraphyletic on the tree. You'll also note that strangely that there is this group that geographically in the east, but it does fall out with the Western Pacificus. That would be this branch right here. So looking at that Hendrickson and Bond reconstruction, that paraphyly does actually line up rather nicely with that Western groups of the samples here that were kind of related to that Apachicus. We can see the same thing here. Those are also Western samples that they used to build their tree.
All right, Eric, that's 10 minutes. But
okay, thank you. If we compare this preliminary work to that of Miller and Coyle, they found that Antrodiaetus pacificus, based off of morphology, might be one of this early diverging lineage for several of the taxa in the Antrodiaetus tree. I have some of those taxa represented here, including things like Montanus, which is also from the Northwest. So is Pugnax, Yisoensis, a Japanese species, and Apachicus, a Southwestern species. Those all do fall out within the tree here with Pacificus as this kind of early diverging group. I have a similar pattern, although again, I do have that paraphyly. So the next step for me is going to be sampling in Canada, as I said, getting more of those Pacificus specimens. Looking at just Pacificus specifically, I would like to understand what's going on with those populations. So I want to look at some of the genetic structure within those disparate lineages. And then finally, where are the boundaries between those lineages? Looking at the Pacific Northwest more generally, I want to continue to refine that preliminary tree of Antrodiaetus. I'm going to do that by including some of the Eastern taxa. Eastern taxa were included in the work of Miller and Coyle, as well as Hendrickson and Bond. And I want to see if that kind of shakes up my tree, if that changes any of the relationships. And I would also like to include taxa described by Koeckendorfer, who described several taxa in the Northwest that I don't currently have represented in my work. So those need to come in there, as well as Antrodiaetus cerberus, which through a recent collecting trip we now have, and that will be included in the data set. I would like to thank UC Davis for hosting our virtual conference this year, as well as the Bond Lab at UC Davis and the Hedin Lab at SDSU for providing samples and just lots and lots of support about a group that they know far more than I do. Hopefully currently, members of the Hedin Lab at UI, so my lab at the University of Idaho, as well as the Vincent Roth Fund for Systematics Research from the American Arachnological Society that's going to be funding some of that field work to be done. And with that, I will take any questions. Thank you for being here.
Thank you very much, Erik, for the very nice talk. I'm getting some questions in the chat. Sebastian Echeverry is asking about the split in the distribution between the North American and Japanese species. So he's asking, how did these species end up so far apart?
Yes, so as I said, these lineages are very old. The work of Miller and Coyle hypothesized that some of these splits go back tens of millions of years. So we're talking about back when there might have actually been land bridges connecting the Nearctic to the whole to the Palearctic regions. But yeah, it is interesting that there is this that the Japanese group does not represent this unique clade by itself. So there, at some point, there had to have been this secondary invasion of Japan by Antrodiaetus. So a lot of this just has to do with how old these lineages are.
Okay, nice. Someone else asked, how would you explain the Japanese species being nested into the North American clade?
That is a good question. It's something that I think is still kind of up for debate, and a lot of that's going to have to come down to timing. There is a fossil species, or there is a fossil not of Antrodiaetus, but of within Adipoides. I think dating is going to be divergence dating is going to be in order to try and figure that out, but I don't have a very good explanation for that. And like I said, I think we're going to need some timing to understand that biogeographical event.
Excellent. Yes, now you're getting a lot of compliments on the chat, and also Catherine Scott is saying that if you can get to Vancouver Island or Haida Gwaii, I don't know if I can pronounce that correctly, but she says that there are a lot of folks who can help you or probably send specimens, and that she finds a lot of specimens near Victoria.
Yes, I think a lot of this work is going to rely on outreach and work and networking. These animals are spread over a very large region. I'm already relying on people helping me out with other labs sending their collections, but yeah, that's going to be a key part of making sure everything's represented in this tree.
Excellent. Well, thank you very much for the answers and for the talk. Everyone's clapping and sending compliments. And now we have to